Srinivas, Department of Biology. In the last class, we studied about the different types of nutrition. In the first topic, nutrition. In that, we discussed in detail along with the examples related to the different types of nutritions. And there are two types of nutrition methods. One is the autotrophic nutrition, the second one is the heterotrophic nutrition. Today we are going to discuss about the autotrophic nutrition, how the green plants and green algae are performing autotrophic nutrition. As you know already, the definition of autotrophic nutrition, in which the individuals are able to prepare their own food materials. That was the information in the last class. Today, how these plants which are green in color performing autotrophic nutrition, the answer is photosynthesis. These plants are performing an important topic and process named as photosynthesis. Actually, photosynthesis is the Latin word. In this photosynthesis, there is a combination of two words related to Latin language. One is called photo, which means light, as well as the second one, synthesis, which means production. Here you can mistake. It's not the meaning that production of light. It's the meaning that production of food material by utilizing light. That is the inner meaning of photosynthesis according to Latin language. So on this photosynthesis, there are so many scientists. They worked hard before 200 years back, and they proposed some statements without any proof. The statements in that the popular one is that the green plants which are living on the planet Earth are the universal producers for the food. After them, after their studies, after their researches, another important scientist just 200 years back named as von Helmont. He proposed, after doing several experiments on the photosynthesis, that the plants are absorbing the requirements not only from the soil but also from the other sources like atmosphere and other media. So basing on his experiments, he proposed one simplified photosynthetic equation in which he given and showed a simple equation. In this, he added the carbon dioxide combined with the two molecules of water, which gives rise to a carbohydrate molecule along with the one molecule of water as well as oxygen. This was the conclusion at last what he found. Before this equation, before this research, he did in a different manner, in which instead of water, he used only H2S gas, that is nothing but hydrogen sulfide. When he used hydrogen sulfide gas in the photosynthetic equation, before this simplified equation, the products formed are sulfur gas but not oxygen. After the von Helmont, after his studies in the year 1936, one more famous scientist in those days named as Robert Hill, he proposed the complete equation for the photosynthesis by utilizing water and carbon dioxide as the reactants. In that, not only carbon dioxide and water, there is also requirement of sunlight and chloroplast. When these four items are combined together in his laboratory, and when he did the research on this, the products which are formed during this equation are one glucose molecule, 6 molecules of water, as well as 6 molecules of oxygen, but the oxygen escapes into the atmosphere. This complete equation first proposed by a famous scientist named as Robert Hill, basing on his work, basing on his interest. This equation was popularly called as Hill reaction. After the Robert Hill, there are so many other scientists like Melvin Kelvin and others did lot of work on the photosynthesis, and they given complete mechanism of photosynthesis. And after knowing about this equations which are related to photosynthesis, the Next important practical work, because science cannot be without any laboratory work. So for that purpose, one activity we are going to discuss. In this activity, we are going to prove that the green leaves perform the photosynthesis as well as they prepare starch. Then how can we prove in a laboratory manner? For this, there are requirements, what we can say the apparatus. In this activity 1, the apparatus which are required are beaker, test tube, one fresh thin green leaf, and lukewarm water, methylated spirit, asbestos gauze, tripod stand, and a Bunsen burner, petri dish, and iodine solution. These are requirements for this activity. Then procedure, next step. In the procedure, let us take one beaker, fill with water, take one test tube, 80% test tube is filled with methylated spirit, in that put the thin green leaf and put in the beaker as shown in the figure, and heat gently by putting on the tripod stand and having asbestos gauze with a Bunsen burner. After few minutes, the water starts boiling, then remove the test tube from the beaker and keep aside gently without any disturbance and handle with care. Later on, slowly take out the leaf. When we boil the methylated spirit which contains green leaf, the chlorophyll pigments which are present in the green leaf are spoiled or damaged, such that the green leaf is converted to colorless or pale yellow in color. So that leaf from the test tube is separated with a small brush or with a small scalpel with a hand with care and put on a petri dish. After putting on the petri dish, you can see the pale yellow colored leaf. On the pale yellow colored leaf, add 1 or 2 drops of iodine solution. When we add 1 drop of iodine solution on the pale colored pale colored leaf, that color is changed to blue black. When the leaf color changes to blue black, then we can understand that the leaf perform the photosynthesis and prepare the starch. This was proved practically, and this is the observation part in this experiment. Then what about conclusion? By doing this experiment, one can say that the green leaves are performing photosynthesis by absorbing carbon dioxide, water in presence of sunlight and chlorophyll, and prepares glucose, water along with oxygen, and the oxygen escapes out through the stomata from the leaf. Is proved practically. Then what are the precautions? Because for a science student, the precautions are very important to conduct an experiment in a successful manner. There are 2 precautions minimum we should take while doing this experiment. First one, the water what we are boiling should be gently and with a less temperature. If you boil more and more, the total life of the leaf will be spoiled. Then our experiment will be failed. That's why you boil the water in a gentle manner with a low temperature. That is the first precaution. Second precaution is that remove the leaf with a carefulness, or else if the leaf is stored out, then our experiment will be failed. So these 2 precautions must be maintained in a clear manner. Then we can get the result successfully, and we can complete this experiment successfully, and we can prove that the green leaves are preparing the starch by performing photosynthesis. Thank you, one and all.